And today I'll be giving an overview of mTORopathies, talking about some of their clinical features and the emerging genetics, with a bit of a focus in the latter half of the talk on our recent paper in Cell Reports. So when we talk about mTORopathies, we're referring to a number of diseases that are generally associated with epilepsy, as well as with abnormal activity of the mTOR pathway. mTOR is a protein that plays a critical role in regulating cell growth and metabolism. And when we think about mTORopathies, kind of the classic mTORopathy is tuberous sclerosis complex, which I abbreviate throughout the talk as TSC. This is a multi-system disease in which patients can present with a variety of signs and symptoms. These can include facial and skin lesions, cardiac and renal tumors, seizures, intellectual disability, sometimes even comorbid with neuropsychiatric diseases like autism spectrum disorder, and then these characteristic brain lesions called cortical tubers. And you can kind of think of a tuber like a small cortical malformation, often referred to as a dysplasia. And essentially what this means is that if you look at a cortical tuber under the microscope, you would see abnormal cortical organization and abnormal cells compared to what you'd see in a normal human brain if you look at it under the microscope. And the most common neurological symptoms seen in patients with TSC is epilepsy. Enrique gave a great talk a couple of weeks ago where she introduced this, but essentially patients with epilepsy will have had multiple seizures throughout their life, and this is due to abnormal electrical activity in the brain. So we've actually known for quite a long time now the genetic etiology underlying TSC. It's caused by mutations in two related genes, TSC1 and TSC2, and the proteins that they encode negatively regulate mTOR, and you can see a model of the mTOR pathway here. And generally, patients with TSC carry loss of function mutations in either TSC1 or TSC2, which means that overall, you're going to end up having abnormal activation of the mTOR pathway in patients who carry these mutations. And currently, we've come to understand that in addition to TSC, there are several additional disorders that can fall into this kind of bucket category of mTORopathies, namely the brain malformations hemimegalencephaly, which I abbreviate as HME, and focal cortical dysplasia, abbreviated as FCD. And in these magnetic resonance images of the brain, the red dashed lines outline the areas of abnormalities. So hemimegalencephaly is a brain malformation that's characterized by abnormal enlargement of most or all of a cortical hemisphere, whereas in focal cortical dysplasia, there's a small region of abnormal cortex, which you can see in the image on the right. And patients with these diseases generally present with intractable seizures, which means that they're not able to be controlled by medications. And because of that, they often end up requiring surgical removal or disconnection of the abnormal brain tissue to attempt to gain seizure control. So until just a few years ago, we didn't really know anything about the genetic etiology underlying hemimegalencephaly or focal cortical dysplasia, but we had a couple of clues that made us think that they might have a similar underlying etiology to tuberous sclerosis complex. So for example, if you look at the abnormal brain tissue from any of these diseases under the microscope, you would see some histopathological similarities. In hemimegalencephaly and in some subtypes of focal cortical dysplasia, you see these abnormal cells called balloon cells, which are illustrated in the bottom image. And in TSC, you see essentially the same kind of cell in cortical tubers and in the abnormal brain, which we refer to as a giant cell. So that was one clue that these might have a similar underlying etiology. In addition, if you look at abnormal brain tissue and look at markers of mTOR activity, in all three of these diseases, you'll see an abnormal increase in mTOR activity in the brain of patients with these diseases. And one thing to note is that both hemimegalencephaly and focal cortical dysplasia are what we refer to as focal brain malformations. And so this means that only a part of the brain is abnormal when we look at it by imaging or by histopathology, as opposed to all of the brain. And so another thing that we wondered is whether FCD and HME might be caused by mutations that affect only a part of the brain, and these are what are known as somatic mutations. So in thinking about genetic mutations that cause human disease, traditionally human genetics has studied the role of germline mutations. And these are mutations which are present in all cells of an affected individual. These include germline inherited mutations, which I've illustrated in this first panel. And these are mutations which are present in a parent and are then passed on through the zygote to the affected child and are present in all cells of the affected child. An example of this can be some cases of autosomal dominant epilepsy, such as those caused by mutations in ion channel genes, where the MRI of the affected child will appear relatively normal. Germline mutations also include what we call germline de novo, or of new mutations. And these are mutations which are generally thought to occur during gametogenesis in one of the parents, so when either the eggs or the sperm are forming. And they're similarly present in the zygote and then all cells of the affected child. An example here is a germline mutation in the gene Lis1, which leads to a smooth brain, or lissencephaly on MRI. An important thing to keep in mind is that because these germline mutations are present in all cells of the affected individual, from a diagnostic standpoint, we're able to identify the mutation by assaying any tissue, including clinically accessible tissues like blood or saliva. More recently, we have realized the importance of what are called somatic mutations in non-cancerous diseases, including neurological and neurodevelopmental diseases. So somatic mutations are mutations that occur post-zygotically, which means that they're present in only a subset of cells of the affected individual. Specifically, the mutation will only be present in daughter cells of the originally mutated cell. If a somatic mutation occurs relatively early in development, as illustrated here, it might be present in multiple tissues throughout the body, including brain and non-brain tissues. And this means that it might still be identifiable from clinically accessible tissues like blood or saliva. In this example, a somatic mutation in the gene DCX means that the cells carrying the somatic mutation will not migrate properly from deep inside the brain to the outer cortex, leading to a double cortex image on magnetic resonance imaging of the brain. On the other hand, a somatic mutation that occurs relatively late in development might occur late enough that it's present only in a subset of cells in the brain. And in this case, it would require direct assaying of brain tissue to identify the mutation, which has important implications for clinical diagnostics, as it would require a brain biopsy. So somatic mutations can cause human disease, including neurological disease, through several different mechanisms. On one extreme, we have what are termed obligatory somatic mutation disorders. And these are generally diseases in which the mutation has only ever been identified in the somatic state, and it's believed that a mutation in the germline state would be incompatible with life or lead to death in the first few years of life. An example of this is Proteus syndrome, illustrated. You can see the hand of a patient with Proteus syndrome in this image. This is a multi-system overgrowth disorder that was recently identified to be caused by a somatic activating point mutation in the kinase AKT1. A second mechanism is what we think of as sort of a second hit mechanism. In this case, the affected individual will have a germline mutation in one allele of a gene, and then a second hit somatic mutation in the other allele will lead to manifestations of the disorder, which are often overgrowth manifestations. So as I mentioned, in tuberous sclerosis complex, generally an affected individual will have a germline mutation in one allele of TSC1 or TSC2. And it's been shown for many of the non-nervous system tumors, as well as in some cases for the cortical tumors, that those result from a second hit somatic mutation in the other allele. And then finally, kind of on the other extreme are disorders where you can find both somatic and germline mutations. And what we've seen recently is that there's often a relationship between the type of mutation and the phenotype severity, so that affected individuals carrying a somatic mutation might have a milder phenotype than those carrying a germline mutation. An example of this is mutations in the gene Lis1. Somatic mutations lead to only neurons that carry the mutation migrating abnormally in the brain, resulting in that double cortex picture on MRI. Whereas if you have a germline mutation, all of the neurons carry the mutation and migrate abnormally, leading to that smooth brain or lissencephaly picture on MRI. So over the past decade or so, our ability to identify germline and somatic mutations has really been greatly aided by advances in next generation sequencing technologies. And these are essentially kind of massively parallel sequencing experiments. So in this example, each gray bar refers to a sequencing read. And the average number of times each genomic location in the target region is sequenced is the read depth. So in this case, we have 10 gray bars at this location, so we have a read depth of 10. For a germline heterozygous mutation, we would expect 50% of the reads to have the mutation. The reference bar at the top shows that the reference base of this genomic location is a C, and we can see that 5 of the sequencing reads have a C and 5 of them have a T. So we would interpret this as showing a germline heterozygous C to T mutation at this location with a 50% alternate allele frequency, which is abbreviated as AAF. For a somatic mutation, we expect by definition that the mutation will be present in less than 50% of the reads. So in this example, 9 of the reads have the reference C, while 1 of the reads has a T. So one interpretation, and this is a true somatic mutation, is that this represents a somatic C to T mutation with a 10% alternate allele frequency. However, we have to be really careful to discriminate a true positive somatic mutation from false positives that can arise, for example, due to sequencing errors. And this is where depth of coverage is an important consideration. We're more confident in a mutation that we see in 100 out of 1,000 reads as being a true somatic mutation, as opposed to one that we see in only 1 out of 10 reads, where it's more likely that that one read could be a sequencing error. And this is also where independent validation in these experiments is really important, kind of to show on an independent platform that you're still able to identify a true somatic mutation. So in order to identify kind of the underlying germline and somatic mutations in mTORopathies, specifically in our case in hemimegalencephaly and focal cortical dysplasia, this was the general model we used. So we have had a lot of wonderful collaborators who have helped with patient recruitment and to get us surgical resection samples from when these patients undergo brain resection surgeries to try to help with their seizures. We're able to extract DNA from that brain tissue and from non-brain tissue when available. And then we perform targeted deep sequencing. We generally focus on genes that are in the mTOR pathway. And the reason for this is that if we have a smaller target region for sequencing, we're able to get a higher depth of coverage for a given cost. And then we use various methods to identify and validate pathogenic mutations. So really the first identification of a genetic etiology for either of these disorders came in 2012 when our lab and then a couple of others published showing that you can have somatic activating mutations in positive regulators of the mTOR pathway in hemimegalencephaly. So in this case, this patient, you can see compared to a normal brain, had abnormal enlargement of an entire cerebral hemisphere, which is outlined in red. And because of intractable epilepsy, ended up having a hemispherectomy where essentially that entire half of his brain was removed. And you can see that post-surgery, that area is just filled with CSF. And this actually did help him to lead a relatively normal life, although there are some subsequent weakness on that side. And sequencing from the abnormal brain tissue from this patient demonstrated a somatic activating point mutation on AKT3. So if you look at the sequencing trace from the brain, you can see that there's a big yellow peak at the location indicated by the arrow, which is the reference or wild type allele. But then in the brain, there's also this tiny red peak that you could easily miss and think maybe that's background noise. We don't see that red peak in the blood. And then when we did a validation experiment using subcloning, we found that this was in approximately 35% of cells. And this work was done by Anne Poduri, who now also has a lab at Boston Children's Hospital and is a pediatric epilepsy specialist. So over the past 6 years or so since this initial paper, we've been able to collect over 100 cases of hemimegalencephaly, focal cortical dysplasia, and some related megalencephaly syndromes. And we've used targeted deep sequencing of mTOR pathway genes to be able to identify a genetic etiology in over 40% of the cases where brain tissue is available, but importantly, in none of the cases where we don't have brain tissue available, which really has important kind of clinical diagnostic implications when we're thinking about being able to diagnose patients that come to the clinic with these phenotypes. So kind of moving a little bit more into our recent paper, we have found, and I should say that many other labs have also been working on this in parallel and found similar things, that focal cortical dysplasia and hemimegalencephaly appear to, in the majority of cases, be caused by mutations that activate the mTOR pathway via a few different genetic mechanisms. So on the one hand, we've been able to identify kind of single hit activating mutations in positive regulators of the mTOR pathway. This includes the upstream AKT3 and PI3K family members, as well as a lot of mutations in mTOR itself, particularly for FCD cases. And this also includes kind of larger somatic structural rearrangements of chromosome 1q, which encompasses the gene AKT3. One thing to note from this table is that in all of the cases where blood was available, you can see that we weren't able to identify the somatic mutation in blood, which really points to the fact that these mutations likely occurred late enough in development that they're limited to a subset of cells in the brain. And the other thing to point out is that if you look at the percentage of cells carrying the mutation, you can see that it varies quite widely. And in particular, you can have as few as 8% of the cells in a hemisphere affected, and that still leads to abnormal overgrowth and function of that entire hemisphere, which really highlights kind of the importance of brain circuitry and how just a few kind of abnormal cells can have big implications for that entire half of the brain. So on the other hand, kind of the other mechanism that we see is loss of function mutations in mTOR negative regulators. This includes the GATOR1 complex of DEPTIC5 and NPRL2 and 3, as well as, interestingly, mutations in TSC1 and TSC2 in a couple of cases that don't seem to show other physical exam or imaging signs of tuberous sclerosis. We're particularly interested in these two hemimegalencephaly cases, where we were able to identify a Twohit model of both the germline and somatic mutations in TSC2 associated with, from what we can tell, isolated hemimegalencephaly. And as I mentioned earlier, this kind of follows Knudson's original Twohit model that was developed for cancer, where an affected individual will carry a germline mutation in one allele of a gene, and then a second hit somatic mutation occurs in the second allele, leading to overgrowth. So the next question that we wanted to ask ourselves after identifying these mutations was to try to understand if there was a particular cell type in the brain that was important for the pathogenesis of these disorders, and to understand if focal cortical dysplasia and hemimegalencephaly might represent discrete diseases caused by mutations in different cell types. And so to kind of get at this question, we used a single cell sorting and sequencing method that was developed by two wonderful previous graduate students in the lab. And this allowed me to sort neuronal and glial cells from cases where we had identified pathogenic somatic mutations. And so in this graph, along the x-axis, we have different cases, and the y-axis is the percentage of cells carrying the mutation. The purple bar is what we got from bulk DNA sequencing, so from our original next-generation sequencing experiments. The dark blue bar is the NUN positive cell population, or the neurons, and the light blue bar is the NUN negative cell population, which would include all of the glial cells. And so what we were able to find was that in all cases, we always see the mutation present in the NUN positive or the neuronal lineage, but it's variably present in the non-neuronal lineage. And so this suggested to us that mutant neurons seem necessary for the development of these disorders. In addition, for the two FCD cases where they had the smallest number of cells carrying the mutation based on bulk sequencing, we also are able to detect the mutation only in the neuronal lineage and not in the non-neuronal lineage, suggesting that in some cases, particularly those where a small number of cells carry the mutation, mutant neurons appear to be sufficient for the development of these disorders. To further investigate the critical cell types, working with two postdocs in the lab who did this mouse work, we developed two mouse models where mutant PIK3CA is expressed that allows for mTOR activation. And this expression was limited to either the dorsal telencephalic lineage, which gives rise to most of the excitatory neurons in the brain, or to the interneuron lineage, which will give rise to those inhibitory interneurons in the brain. And what we find is that when we have mTOR activation in the excitatory neuron lineage, we see dramatic brain enlargement, which is referred to as megalencephaly when the entire brain is enlarged. And we also see abnormal cortical lamination, especially in those upper cortical layers. On the other hand, when we have this mTOR activation in interneurons, we do not see any significant difference in brain size or in cortical lamination. The only defect we were able to find was a slight decrease in the number of interneurons that migrate to the cortex. And so from this, we think that the dorsal telencephalic lineage, and in particular the excitatory neuron lineage, is really critical for the development of these phenotypes as opposed to the inhibitory neurons. So our data together to us really suggest that focal cortical dysplasia and hemimegalencephaly don't represent discrete diseases, but rather represent a continuum of disease caused by somatic mutations in these dorsal telencephalic progenitors. So in this graph on the x-axis are individual cases where we've identified pathogenic somatic mutations. On the y-axis are the percentage of cells carrying the mutation. And then the MRI images go in order from top left, from left to right, in terms of the percentage of cells carrying the mutation. So you can see that in general, as the percentage of cells carrying the mutation increases, there's a trend towards a more severe phenotype, but there still remains overlap between what we can classify as focal cortical dysplasia and hemimegalencephaly. We can see this kind of correspondence when we look at individual alleles. So this is kind of one striking example where if we look at the amino acid C1483 and mTOR, where there's been recurrent mutations identified, we can see that in one case where about 15% of the cells carried the mutation, it led to a focal cortical dysplasia. In another case where up to 70% of cells carried the mutation, it led to hemimegalencephaly. And then finally, we had identified a case of a de novo germline mutation at this amino acid. So this mutation was present in all cells and was identified from the blood. This led to bilateral brain enlargement or megalencephaly, and unfortunately was not very compatible with life. So this resulted in a multi-system disorder where the boy unfortunately passed away at a little over 2 years old. On a bigger picture, we also performed a literature review, kind of looking at all of the reported somatic mutations for these two diseases in the literature, and we found that overall, there is a significant difference where the alternate allele frequency reported for somatic mutations and hemimegalencephaly is significantly higher than that reported for focal cortical dysplasia, but we still see an overlap between the two. There's not a dividing line where you can say, okay, if this many cells carry the mutation, it's definitely going to lead to hemimegalencephaly versus focal cortical dysplasia. And kind of like overall, we believe that the lesion differences that are seen really reflect the developmental time and original progenitor cell where the somatic mutation occurs. So starting kind of at the earliest time point, if you have an activating mutation in an mTOR positive regulator that occurs before fertilization, so this would be a germline mutation, it will lead to bilateral brain overgrowth and a lot of systemic manifestations. And this will generally be lethal either in utero or in early childhood. Such as the case I just discussed of the young boy who had a germline mutation in mTOR and had megalencephaly and a lot of other systemic issues. However, this mutation will be identifiable in all tissues of the body, so you can find it in the brain, but you'll also be able to find it in a clinically accessible blood sample. If a mutation if an individual has a two-hit set of mutations where they carry a germline mutation in one allele of a negative regulator of mTOR and then develop a second hit somatic mutation later during development that's limited to the brain, this can also lead to kind of a focal brain malformation. And in this case, one of the mutations, the germline one, would be identifiable in both blood and brain, whereas the second mutation that occurred later in development somatically would only be identifiable in the brain. And then finally, you can also have single-hit activating mutations that occur somatically later during neurogenesis in positive regulators of the mTOR pathway. And depending on whether these occur earlier or later in development, we think that these are more likely to lead to hemimegalencephaly and focal cortical dysplasia, respectively. But these mutations, again, would only be identifiable if you're directly assaying brain tissue. So in conclusion, I think the big takeaways here are that when we think about mTORopathies, the main diseases that we're thinking about are tuberous sclerosis complex, focal cortical dysplasia, and hemimegalencephaly. Generally, most patients with these disorders present with epilepsy, and we think that the main underlying etiology is mutations that activate the mTOR pathway. We're very hopeful that identifying these mutations in FCD and HME kind of points to the promise of using mTOR inhibitors, which are currently being studied in clinical trials for patients with TSC, and also using them in patients with FCD and HME. And if they're effective, then these children might not have to undergo kind of drastic surgical hemispherectomies or functional disconnections. And then kind of more broadly, the time and place that a mutation occurs really is critical for determining its consequences in the human brain. And from a technology standpoint, next generation sequencing experiments have been very useful for helping us identify germline and somatic mutations in neurological diseases. So I just want to end by thanking my PI, Chris Walsh, who's really been a wonderful mentor, as well as Anne Produri, who co-mentored me on this project, many members of our lab and collaborating labs who's helped with various aspects of this project over the years, a lot of neurologists, neurosurgeons, neuropathologists who helped us with patient recruitment, samples, and interpreting imaging, our core facilities and funding. And then that's a picture of our lab going to a Red Sox game this past summer. So thank you. I'm happy to take any questions.